Hello everyone, welcome to a brand new video where we're going to discuss mitosis. So what we are going to look at in this video is seeing the three major types of cell division, what the uses are of mitosis, what about three major purposes for mitosis in general. Then we want to look at our chromosome structure, review the cell cycle, and then look at what happens at each step and what goes on through mitosis. So as we really begin, I really just want to talk about that there are three major types of cell division that's out there, and they are unique for different purposes. So we are going to discuss them. And the first one, which we're not going to discuss in this video, is called binary fission. Now, other than a very short kind of discussion, binary fission is found in prokaryotes and what you might think of as some lower protists. So that would be things like your single-celled eukaryotes who are a little less complex there. Then we're going to see mitosis, which is going to be our focus for this video, and then meiosis. Now, one of the things I want to talk about is mitosis is where we take a cell who has two copies of each chromosome and create cells with two copies of each chromosome. So we would say we would go diploid to diploid, or we also could go haploid to haploid, one copy of each chromosome to one copy of each chromosome. Now, meiosis, this is primarily going to be used in sexually reproducing organisms to make gametes, things like sperm cells or an oocyte of various types. And this always goes from a diploid cell to a haploid cell, meaning that there's two copies of each chromosome down to a cell with one copy of each chromosome. Now, I'm not going to spend a lot of time talking about that. That will be something I'll do a different video on. Now, what we want to do is about why mitosis? Why do we do it? Why would we need to do mitosis? Well, there are three big reasons why cells undergo this kind of cell division. And I'm kind of grouping the first two together. One of these is either a growth, which means when you want to grow, you had to grow by increasing the number of cells that your body had. You had to increase the number of bone cells to make the bones get longer. You had to increase the number of muscle cells, things like that. You had to increase the number of skin cells. You began as one cell, and you began to be trillions of cells. So we had growth and also used in your development. What happens in the early stages of development is rapid cell division. So growth and development is the first major things. I'm going to put these kind of together instead of separating them. It's just say growth and development. Since they kind of go together, after you're done developing, you will continue to grow. Now, another thing is just pure replacement. When you have cells, let me try to redo that. When you have cells that have died and you need to replace those cells, replacement of damaged cells, replacement of older cells if they're damaged or old. And then also for some organisms out there, purely for their reproduction, you think about your protists out there, they need to reproduce, they use mitosis, or they're going to use some intermediate between mitosis and binary fission, or they might use binary fission. But reproduction for single cell, for one cell organisms, they would do that. So as we think about that, that's some of the main reasons why. Now, what I want to focus on here is discuss a little bit more about eukaryotic cells, their chromosomes, and what's going on. So if we were to look in a normal eukaryotic cell, let's say this cell membrane of a normal eukaryotic cell, and I look inside that cell, somewhere in that cell, there will be a nucleus. And the nucleus of that cell, we know, contains the DNA. Now, DNA is in various forms, but when the DNA is dividing, we know it's in the state called a chromosome. Now, these chromosomes, what happens with your chromosomes is you are a cell that has 2 copies of each chromosome in your body. Your somatic cells have 2 copies. So what do we call it? If you have 2 copies of each chromosome, we refer to that as being diploid. Now, a diploid cell has 1 chromosome they got from their mom and 1 chromosome they got from their dad. And so I've got the maternal chromosome in red here, the paternal chromosome in blue here. Now, all chromosomes have a few major things in common. What I'm going to do for each one of these is put a dot in the middle of these chromosomes just to make things a little simpler. So we want to start off with first is what are these red lines? The red lines that I'm pointing to here of each chromosome and the blue lines on each chromosome, this references something called a chromatid. What is a chromatid? That is a string of DNA. That is a DNA strand wrapped around some proteins. Usually what you're going to see with chromatids is that your DNA molecule here would be wrapped around a group of proteins. And if I were to draw 8 of these little proteins here, there's 4 of them. Here's the other 8 on the other 4 on the other side. You would have these 8 proteins, and you would have a DNA strand that just wraps around it here, and that would make up your DNA and your these little proteins called histones, and this creates a nucleosome. And what you have in that nucleosome is these guys coil up nice and tight, tightly condensed to make a chromosome. So each chromatid here is what we refer to as being a DNA strand. Now, I do notice here that on the red one there are 2 chromatids here. Notice that there's 2 chromatids. These chromatids are called sister chromatids. Sister chromatids. That now is very important. They are genetically the same. They are the product of DNA replication. So they are DNA replication products. Now over here we would have another set of sister chromatids here on the blue chromosome, on the paternal chromosome. So that those would be, both of those would be sisters as well. Now, these sister chromatids, the 2 red ones, are identical to each other, and the 2 blue ones are identical to each other. But if we look at the red one and the blue one, they are not the same. As the red one comes from your mom and the blue one comes from your dad, and they are not the same person, you are seeing 2 genetically distinctive individuals. These are referred to as homologous chromosomes. And homologous are different. And that is a very important thing to know about the difference between sister chromatids. Sisters, same. Homologous, different. So homologous chromosomes are a pair. So when you have a diploid cell, they will have homologous pairs. There will be one maternal from your mom, one paternal from your father. Now, also what you will see about them is, let's look at each one of these a little bit more uniquely. Let's take one here. Let's take one chromosome and draw him out a little larger so we can see a little bit more details about one of them. Each chromosome here is attached in the center, a center part, an area where there's these cohesin proteins that are holding them together. And these cohesins that hold it together are at this thing called a centromere. And the centromere holds the 2 chromatids together. Now, on each chromatid, there is a region of a chromatid that sticks out past the centromere. These are referred to as chromosome arms. Now, usually for an arm, there is usually a short arm oftentimes called the P arm, P for petite, and a long arm, and we refer to that as the Q arm for chromosome. And at the centromere, there will be these protein areas that are there for microtubules to bind. And these microtubule binding areas here that are on that, these are referred to as the kinetochore. Kinetochore proteins are for microtubules bind. This is where microtubules bind to help in division. Now we're going to talk about that. So it's definitely important that we pay attention to chromatids, homologous pairs, sister chromatids, things like that, your centromere and kinetochore. Those are going to be the most important things for what I'm going to talk about next. Now, when we talk about cell cycle as an overall thing, basically this is a cell's life cycle. Now, with cell cycle, what I want you guys to be thinking about is imagine a clock. If we were to think about a clock, and let's imagine this was 12 hours of a day. We're not going to all be talking about a 24-hour. Let's imagine a 12-hour clock here, because the clock goes around once for 12 hours, goes around again for the 24 hours in a day. So we're going to look at it think about it as in a 124-hour time period. We're going to slice the clock up into 3 major slices. And these 3 major slices, we're going to come into that one slice and we're going to take another piece out of that. So now we got 3 major slices and 1 slice we're going to take a big thing out of. Now what we will see is everything from here to here which would be about, if we were talking about a 12-hour clock, about 90% of that time would be what we call interphase. So this would be all interphase. Then you have only this small part here is the mitosis phase. And those are the 2 major phases of what is referred to, guys, as your cell cycle. Is a lot of interphase, and then what is referred to as your interphase. Now, mitosis and interphase. Interphase being the longest. If you were to be thinking about that in terms of time, it's roughly 90%, roughly there of overall time spent. So 90% of 12 hours. Now, mitosis itself has got a few smaller divisions. P, M, A, and T. PP mat. Then we're going to see a C. And we're going to talk about those a little bit more in detail, but we're going to think about PPMAT. Now, so as we think about mitosis proper, we'll get into what those mean. So as we got our pie drawn out, let's look at what happens during interphase and what happens during mitosis phase. Interphase is made up of 3 distinctive phases: a G1 phase, an S phase, and a G2 phase. These are subphases of interphase. Now, to remember all of these, you have your mitosis and interphase. Interphase, think of go, Sally, go, make, mitosis, make, cytokinesis, children. Go, Sally, go, make children. This is cytokinesis. And I thought I could write that in there. So definitely not going to work very well for me. So what's about that cytokinesis? Go, Sally, go, make children. Now, G1 has got a lot of important things that happen during G1. G1, you've got protein synthesis. A lot of protein synthesis happens here. A lot of protein synthesis. This is where you're going to try to produce proteins for a cell getting ready to divide. You make new organelles. That's one of the things that's going to happen here. Make new organelles because cells getting ready to divide. And so that's like when your mitochondria and your chloroplasts are going to divide. And that's when the cell grows. Cell gets bigger. It enlarges. We increase the size of your cell. It actually doubles. Cell actually doubles size. Cytoplasm doubles size. S phase is when DNA replicates. So the synthesis of DNA. DNA replicates, DNA synthesis. S phase, think of it as synthesis of DNA. G2 is mostly growth. In prep for division, mitosis. Now, mitosis goes through the stages of PPMAT. Prophase, Metaphase, Metaphase, Anaphase, and Telophase. Let me write these down a little bit neater. So, for mitosis phases, you have P, M, A, T, PPMAT. And so I always tell my students, imagine you got a naughty little puppy named Mitosis, and he goes to the bathroom in your house, so you got to go get a pee-pee mat. Prophase, Metaphase, Metaphase, Anaphase, Telophase. PPMAT, PPMAT. Okay, so these are what we're going to be focusing on. We're going to look at what happens. Now, at the end of mitosis, you're going to enter a process called cytokinesis, and I'll talk a little bit about cytokinesis a little bit as well, getting into that. But we all know before the mitosis becomes interphase. So let's focus a little bit about interphase here. What is happening during interphase is cell is getting ready to divide. So if I were to look at a cell that's getting ready to divide and I were to look at its nucleus, the DNA would not be coiled up into chromosomes. It would be a loose association of DNA. That is just uncoiled DNA that we refer to as chromatin or chromatin. Chromatin is non-dividing DNA. And that is also a very important thing. If I could write here, sometimes it is harder than I would think to write with my stylus. This is non-dividing DNA. Non-dividing DNA is the chromatin. So it's in this nasty little kind of mix. Now also what you're going to have is during this part there will be an area here where there is a pair of centrosomes. So we have a centrosome, a centriole pair. So a centrosome, you have one centriole here and a centriole there to make a centrosome. So during this, the centrosome is kind of beginning to produce some microtubules that'll be here. So you will see microtubules present during this stage. Now what we are going to see is after interphase, remember interphase is getting ready to divide. So this is where things get really interesting and where we really want to make sure we understand what's going on. And that's going to take us on to our next stage, and that's going to take us to prophase. Prophase, let's break this down. First off, pro meaning first. So this is the first phase. Prophase, first phase, first step. Now, the hallmarks of this step are going to be one of the first things I want to make sure we all pay attention to is that first off, your nuclear envelope, the membrane around the nucleus is intact. It is present. So that's the first thing we're going to notice. Second thing you're going to notice is the chromosomes have condensed. So we want to make sure we notice that there are here in prophase condensed chromosomes. That is also a very important thing happening here. Chromosomes have condensed. The other thing you're going to notice is, sorry, this was supposed to have been in green here, is that my my little centrosomes, they are beginning to separate here. They are separating. So you're going to see this, what is basically called mitotic spindle, really starting to form up. So this is what we're actually starting to see is the mitotic spindle is forming. And so basically your centrosome pairs are separating. Now a mitotic spindle is you got these centrioles that produce microtubules. So there is a centrosome, remember, is two centrioles. Once we separate these centrioles, you have your aster, the microtubules that come off, will be associated. Your astral rays, your radial array of microtubules. And they begin to separate, and then now these will get replicated during interphase. One of the things that happens during interphase is replication of organelles, and that's one of the organelles that's going to replicate that you'll have extra copies of. Now, with this beginning to happen, that's going to take us into our next stage. And our next stage, Prometaphase. So this tells me this is before Metaphase. Okay, before Metaphase. So we know this should be before Metaphase. Now we have our cell here. First thing that I want to point out that you're going to see happen is that the nuclear envelope breaks down. So that's the first thing I want to talk about is nuclear envelope breaks down here. Okay, there is no nuclear envelope. And because there is no nuclear envelope, our chromosomes, our homologous chromosomes, they are floating around free. Now remember, we talked about the centrioles separated. Now they're going to be at the opposite poles. And the microtubules are going to come in and bind to the kinetochores. So nuclear envelope breaks down, and spindle fibers attach to the kinetochores of the centromere. Okay, so hopefully that's readable in my drawing. It can get really kind of difficult sometimes to draw with these stylus, especially it's kind of cold in my office today, so it can be a little more difficult. All right, so prometaphase, we have that happening. Now what's going to be the next phase that we're going to see is going to be metaphase. Now metaphase, there's a few things I want to talk about real quick. Meta, we're going to be meeting in the middle. Meet in the middle. And that's I always like to talk about metaphase. This is when you meet in the middle. This is the meet in the middle phase. Now remember, nuclear envelope broke down already. So since the nuclear envelope broke down in prometaphase and our DNA strands are floating around, what I want you guys to do, if you're drawing with me, is to take a dotted line and draw it right down the middle here and label this dotted line the metaphase plate. This metaphase plate is an imaginary line, but what happens is your chromosomes line up at the metaphase plate, where each sister chromatid is facing on each side. So you have sister chromatids on each side. And remember, these little centrioles here, they are attached to each of the kinetochores there. They are attached to kinetochores, and they've lined it up. The chromosome pairs align on the metaphasic plate. Now, after metaphase, our next phase that we're going to see, remember, is anaphase. Now, in anaphase, this is our sad phase. This is very sad phase. Now, we had lined up our chromosomes at the metaphasic plate. And each of these microtubule secreting things are the astral rays, the mitotic spindles, were on each side. And what they've done is, when they grabbed a hold of your sister chromatids for each chromosome, they grab it and they pull them apart. They begin to separate these two. And what is here that's so important to make note of is sister chromatids separate. And that is the main event here. Sister chromatids are separating. We separate the sister chromatids. Now that we've separated these chromatids, we can now go and put these chromatids into separate cells. But we got to take this big cell right here, this cell right here, and make it 2 cells now. So how are we going to do that? There's a process called telophase. Telophase, remember PPMAT, PPMAT, telophase, telo equals far away, far apart. Okay? These are going to be far apart. So what we're going to do is, right there where we had our metaphasic plate, let's imagine now that I've got this going down. And we have this little thing in the middle here going on called a cleavage furrow. Cleavage means it's cleaving in half. It's beginning to take and pinch the cell in half using these contractile rings of proteins that will begin to pinch it in half. And because we have a chromatid each inside this, we now have the nucleus reforming. Nuclear envelope reforms here. And it's going to be enclosing your chromosomes, that your sister chromatids you separated. So now that we've separated those out, we now have 2 separate cells that are ready to divide apart. So PPMAT is what we saw. And we have PPMAT, Prophase, Prometaphase, Metaphase, Anaphase, and then telophase. At the end of telophase will come the very last step called cytokinesis. Cytokinesis is cell divides. It separates a cell. We have a cell here, we have a cell here, and their nuclei, remember, are containing my chromatids that I had. And we have 2 diploid cells. These cells are diploid because they have 1 copy of each chromosome. They're just non-replicated chromosomes. Now remember, interphase, what's interesting, what was important about interphase we talked about, is that interphase had the Ssubphase. During Ssubphase, a cell here will replicate the DNA, producing chromosomes that are fully possessing two sister chromatids. And that's just being made by these chromosomes here. You're just taking the chromatid and copying it and making full-on replicated chromosomes. So these are replicated chromosomes, very important, and these are non-replicated chromosomes. So that was an important distinction to also make. So if we can remember that overall with what you have in mitosis cell cycle was go Sally go, that is interphase, G1S, G2, make children, mitosis and then cytokinesis, and that mitosis is made of prophase, prometaphase, metaphase, anaphase, telophase. At the end of that, cytokinesis happens. Then you want to go back and look at what happens at every stage and draw it with me. It helps you to better understand if you draw it. We always say sketch it to catch it, draw it to know it. And if you can do that, you will surely master exams where they ask you to do this on things like the MCAT, etc. So this concludes my video on mitosis. If you would like to see me do other videos for the channel, please let me know. But don't forget to comment, like, and subscribe. Let me know what you want to see. Let me know what I do right, what I do wrong, if I can help you in any way. And give me ideas for future videos you would like to see. Thank you guys so much for watching.